Here you are. Ah, happy thing. I've got animations to show you today, about four of them. Yeah. I've got animations to show you about four of them. Thank you. Thank you. Thank you very much. All good. I knew I'd get your attention. I don't have it anymore. But we digress. Now, okay. This is it. The way to frame this. This is just information that you can look back and listen to for this take-home thing I'm going to give you to help grades over the break or the week you come back from break. We're not going to have a tremendous amount of urgency about this. So it's one, if I have 25 questions, the idea is to get all 25, which I am confident that you're going to be able to do. Or else I'll be very dissatisfied. You're clicking. Okay. So this is the Nestor PowerPoint, and this is where that God, there's more noise here.
I'm sorry, I was trying not to be loud.
He can't help him with that stuff. Open it prior to being here. It's okay. I have one of those things that you can't hear. I mean, drinking beer this early is bad.
Oh my gosh. Thankfully it's not here.
We're not that sure about that. It's okay. I have those little cans at home that the ones that have they look like they're whatever they're called. They have like either vodka or something. No, we don't drink them. It's like the kids bring them over for a party, and then leave in the garage downstairs, keep them cold, come back. Oh, children, I forget even get the names. Not that I was expecting anybody here to know any of those names. Now, on with the show. Spontaneous mutations, all those things I was talking to you about prior to this. Okay, we're spontaneous. Let me go backwards. Okay, when I was telling you about the different kinds of mutations, and we're going to look at that. So let me okay, here we are. Spontaneous mutations, normal. So you can go all the way from living things that can occur as rapidly if you without doing the scientific notation. 1 out of 10,000 to 1 out of a trillion. Okay, that's just normal. Bacteria are down 1 in 10,000, 1 in 100,000. They mutate a lot anyway because they don't have the safeguards that I was telling you about the last time we met. Our cells, to the order of maybe 10 million to a trillion, very infrequently, when we are exposed to radiation or noxious chemicals, not just us, but bacteria, that rate can increase by a factor of 1,000. We call that an induced mutation. So imagine for bacteria that 1 in 10,000 could be as frequent as 1 out of 10 replications. Think of that. Now, you're going to see a lot of the things they expose bacteria to in the laboratory to induce those mutations, or things that we would find as what we call mutagens. Nasty things, nasty chemicals that you don't have to remember any of the names about. But one of the chemicals people don't think about that bacteria get exposed to that effectively is chemical warfare is an antibiotic. Understand, it's a noxious chemical to bacteria. Obviously, our goal is to destroy them. But one of the things that it will do is it will mutate them. That is part of the problem. And that will lead, when we get into the unit on antibiotics, to understand if we don't kill them quickly, they mutate. Remember that bacterial growth curve thing? Who are the ones who are surviving when they get to stationary? Why I ask you about this stuff, that stationary phase stuff. Those are the ones that are already a lot harder to destroy. They survive. They mutate, they adapt, they overcome. They sound like the Marines. Okay? Pardon me. So that is what I'm talking about, why this is important. And don't forget about this. This is called reversion. This is called a backwards mutation. It mutates back to normal, rarely. That becomes important when we get to something in today's presentation called the Ames test. These are stuff that you definitely have to know going forward. Okay? So mutations, not anything. Single mutations are comparatively rare, and that's why We use different antimicrobial medications, sometimes 2 at a time. The chances, and we do this with tuberculosis mandatorily. You always have to use 2 antibiotics. We routinely don't do this. Okay. But when we're dealing with ones that we know are extremely resistant to start, okay, they have a major tendency. That's why we have to use more than one. So just understand there's a lot that many of you will see clinically. I know that doesn't include everybody, but it's important to understand this. So when we talk about what I was talking about, are what we call point mutations, this isolated mutation, effectively. And again, you made me go into silent, means basically it doesn't change it at all. Typically, remember, it's always in these 3-letter combinations we classically call codons. Thankfully, we don't have to know about that. Okay, not going to go crazy about it. Missense, incorporating a different amino acid. Typically, it does not function. Again, the sickle cell hemoglobin example is certainly a good one. Versus nonsense, typically they encounter what's called a stop codon. Okay, and that's bad. Those are isolated spontaneous mutations. We're not even talking about induced yet. What happens when other things in the environment, all those nasty reactive oxygen species we've talked about at length, the O2 with the minus sign, superoxide radical, all the bad things you get from stuff that you should do or shouldn't do. Again, whether it's smoking or drinking or ingesting, various kinds of harmful substances or harmful substances in your environment. One of the things they do is they increase the number of exposures to this. And then all of a sudden we see some pretty severe ones. Okay. And that's these things. We see little things that get chopped out. So what happens is you either add a pair or delete a pair, and we call that a frame shift mutation. They read in little 3s. And so what happens, this whole reading sequence gets altered significantly. So all of a sudden, it's going to be, and inevitably, you're going to encounter a stop code. Cowan, and I don't think they have it now. They used to have a really good one that had like a simple sentence like you and I would get in kindergarten or first grade. It was the fit cat blank blank blank, and all of a sudden it became the fat cat, and all of a sudden it became 3 words you couldn't understand because it changed that 3-letter frame. All of a sudden it was it was Hef, H-E-F, because the knocked out the T. All of a sudden the next 3 letters were something like that. Total misunderstanding. That's the difference between a missense mutation, fit for fat. Maybe the cat should have gone to the gym a little bit. Probably looking at our cats at home who eat too much. Okay. They move very slowly unless food is involved. And they're jumping. The one who inhabits the bedroom. It's like that. But you get the idea. That's a frame shift mutation. And moreover, you hear some of the irregularities genetically, where we have pieces of chromosomes missing, or chromosomes out of place, trisomy 21, Down syndrome. Okay, now all of a sudden you have a whole mess that's there. Now it really affects, and I think it's that chromosome 21 is associated a lot with aging. And if you didn't know it, in Downs, it really has been a vehicle to understand, because Down syndrome children and now adults age much more rapidly. That's why their lifespan is often shortened, despite whatever the mental implications, which are always there in any event. But if you look closely, you'll see the typical signs: graying hair, loss of hair, etc., etc. All those kinds of things. So it's very interesting. Okay, when you really want to look at genetics, it's a really interesting field. That we really are beginning to understand much better because of the molecular biology. Okay, that was part of it now. Just to give you some context for all of this. So that when we see these, there's problems. So losing portions of a gene or a gene itself, really bad. Okay, and then these are the knockout mutations we were just looking at. I'll show you the transposons. That's what I was talking about. The animation is better. I'll show that to you at the end of today. So I don't want to, a lot of this stuff is just genetic stuff that I don't think you really need nor would be tested on in a typical program, typically geared to nursing and various kinds of allied health fields. Here are the induced mutagens. Some of these chemicals are wild. We use them in the lab. And look what they do. They alter the nucleide bases. When you're going to hear about that, base analogs are the foundation of antivirals. We use base analogs all the time. If you know somebody who has to take Valtrex, which happens to be a great medication, and I'll explain to you why when we do antivirals, it's a base analog for adenine. That's the foundation for cancer chemotherapy, the most original ones that were there. Intercalating agents are something we utilize in the laboratory all the time. We use something called EDTA. Did you ever hear of that? Anybody? Okay, EDTA is something that leaches calcium out. And we use it a lot when we don't want blood to do certain things. So we pull the calcium out of it when we're doing, like it'll be in one of those different color tubes. Just for context, radiation, thymine dimers, ultraviolet light, that's important to remember. X-rays, actually causing DNA strands to break, hence deletions. Ultraviolet light, we can handle really easily. They go into it in this power. When you and I are subjected to ultraviolet light, okay, it makes these thymine dimers. However, continued exposure to even just in regular light, if we get away from the excessive ultraviolet light, we have mechanisms that repair that. We have a lot of mechanisms. Bacteria have very few. That's why it's so problematic for bacteria and why we utilize it in various industries and in various medical facilities and water treatment facilities. Okay, bacteria have a hard time dealing with those thymine dimers. Thankfully, we do not. Obviously, over time, excessive amount of ultraviolet light is going to cause significant problems over time with DNA. And it's normally in people who are, as they get older. I mean, you might remember me talking about it a little bit to your class when we talked about skin and things like that. You know, it's really down the road that we see the accumulation of these problems for people who have been exposed for long periods of time vis-a-vis basal cell and squamous cell carcinoma, for instance. So these are some of these agents, and most of the time we use them in the lab. So I don't see any need to go through alkylating agents, base analogs specifically, but this just gives you an idea, okay? The wrong nucleotide is incorporated, and what that does is it impedes DNA. That's why it effectively creates the kind of mutation that's incompatible with that DNA being functional. This is one of the early ones. Here's thymine. Here's uracil. It's all about shape. So here's thymine. They take out this side chain, which is a methyl side chain. That's what you have to know that. And place it with a toxin called bromine. Okay, bromine is over there. Halogens, fluorine, chlorine, iodine, bromine. When you put those with something, they're bad. Okay, we usually old days we used to call them fluorocarbons. Okay, nasty. So they could have used fluorine. They happened to use bromine. What we did early days, they used fluorine was one of the original treatments for breast cancer. 5-fluorouracil. It was actually one of the original. I was hearing about that when I was in medical school 50 years ago. We were having this talk about anti-cancer agents from one of our guest lecturers. I can remember like it was, you know, relatively recent. Can't remember his name off the top of my head. Sorry. So here we go with more of that. And these are the intercalating agents. They get in between. Just to give you some context, this is the ultraviolet light one. Thymine dimers. That, gang. Thymine dimers. Nice. Thank you. Okay, ultraviolet light, thymine dimers. Effectively, this is an interesting covalent bond. And if you look at the schematic here, what it basically suggests is this. Okay, it creates an irregularity in the DNA strand. If you look at this, what they call the sugar phosphate backbone. Again, and if you think of the DNA and the best idea is the zipper, it's like having a zipper that's got a defect in it. You can't unzip it or rezip it effectively. So effectively, using the term again, it loses most of its ability to do what it does, as opposed to breaks that can't be repaired, particularly double-stranded breaks. Okay, really significant. And again, I'll show you the transposition one later. So this is cancer. Enormous amounts of spontaneous and mutagen-induced damage, if not repaired, leads to cell death and cancer. For example, two genes, these are the breast cancer code for DNA repaired enzymes. They are what I remember I showed them on the board, tumor suppressor genes, because they have particular enzymes that repair DNA. I can't make this any more significant in discussing those two genes. Okay, high probability of breast cancer and ovarian cancer. This is the determining factor when horribly they discover breast cancer, which I tell you that right now, I think statistically it's 1 in 7 women get that diagnosis in some way, shape, or form. It doesn't all mean horrible. Men, prostate cancer, a little bit higher, but not a whole lot. A little bit lower in the sense 1 out of 8. I mean, these glandular cancers are going to impact many of you. Let's do the numbers. See how many people are in the room. What does that mean? 4 of you are going to get one of these kinds of cancers? Be aware of that. And so I take it's not so much the precautions, but the steps to recognize early detection and treatment. That's the whole ballgame. Find it early, but those breast cancer genes are scary. Oh, gosh. I always tell the same anecdote because you don't have me for doing repro physiology, repro and that, that particular unit toward the end of AP2. My cousin, they had 5 kids. She had mom for 5 kids. Very athletic runner, very slender. They discovered breast cancer. But 2 of her aunts had died of it, and I think her mom. And even though it's not necessarily familial, but she'd elected to have a double mastectomy when they discovered it. And when they did the reconstruct, it didn't work well. One side worked fine, the other side, went a year or so with problems. Really significant. I think they almost lost her. To give you an idea of the severity of this condition and even the follow-up treatment if people elect to do that. Just saying. To give you an idea of the severity of this. Okay, and here are all of these different things. We talked about mismatch repair. Okay. Different kinds of DNA polymerases. They're not all the same. Again, mismatch repair at best 50-50. Is likely to repair it without me walking up and down the aisle again. Is likely to repair it as it is to take away something's mismatched. It's as likely to take away the good one as is the bad one. Where that DNA polymerase III much more significant, much more significantly effective number. That's there. And they talk about these. So again, the proofreading by DNA polymerases, much more accurate in nature, inserts the correct nucleotide or sequence. And again, nothing is perfect. So the whole idea when we're talking about mutations in us, even without thinking about microbes and viruses and things that would cause it, is really avoidance of things that are potentially carcinogenic or mutagenic in nature. It just is. Again, you're listening to somebody who smoked for 40 years. You can't escape it. There's always a complication. Most likely been lung cancer. Mine was bladder cancer. We went to a visitation Monday night after class. I mean, and my inquisitive mind is, he died of complications of bladder cancer. I didn't know him, but my wife and the widow were very good friends. But I didn't know the gentleman. They know each other for quite a long time. I'm sitting there. Was he a smoker? Was he this? Was he? I want to ask that question. It's inappropriate to ask it there. You can't get clinical in those kinds of times. You just got to be. So moving on. That's there. So they're showing you how it fixed. And I used to teach that. People were very disgusted with me trying to go through these slides and explain what was called restriction and modification enzymes. tell you about them in passing, though you don't have to know it. One of the things that facilitates mismatch repair, you got your feet in it. I'm gonna pull that out. But you were leaving out. Those socks, wow. It's all right. I only wear the little ankle socks, so there's always skin. I stopped. Hey, you can be. My wife's looking at me this morning, going, "Jesus, you go to school looking like that." I said, "I get over it. You can be a little eccentric, idiosyncratic, odd, weird. Those are synonyms. Those are synonyms. Mismatch repair. Bacteria, and I said this before, I just want to bring home the point of why they're so important. They make these enzymes, and the way they repair themselves is they make what are called restriction enzymes. They chop up DNA reliably along certain sequences. It's a fancy term, it's called palindromes, little repeat sequences that are there. That's the foundation of mismatch repair. That's also the foundation, I told you this early on in class, of why we can do it. Chops up DNA reliably. And the only way we can analyze DNA, whether it's a paternity test, whether it's 23andMe, whether it's a crime scene, is because of these restriction enzymes. That's the magic of bacteria. They gave us the ability to use these very unique systems that they have to make our lives, if you want to consider it easier or not, I don't know. But genetic, again, the whole idea of us being able to do genetically modified organisms, to make biological things, to make human insulin, to make pitocin, to make antidiuretic hormone, are all because of bacteria. It's from this unit. And I used to teach a lot about it, but I mean, it's more. It's wrong to teach things that I find interesting that you don't necessarily need to know. You have enough on your plate. I think that's I've learned that over the years I've been doing this, but I have fun telling you about it. And so this has to do with the way they repair thymine dimers, light repair and dark repair, and SOS repair. But the problem is, okay, when they do these last ditch repairs in bacteria, why it causes a problem, they don't have any proofing. This is a very unreliable mechanism. Not that I would ask you about it. This is what we do in the lab to grow them. So you did the antibiotic lab this week, did you? Or, yeah. Put the little discs on, all the thing. Kirby Bauer, was that a name that was mentioned? See, I remember this stuff. Effectively, if we want, a lot of times we'll put antibiotics in the agar itself with things like this, okay? Medium containing streptomycin, medium without, to see if it's resistant or sensitive. If it's resistant, they'll grow. If it's sensitive, etc., they won't grow, that type of thing, okay? Medium containing streptomycin, etc., we begin to look at those things that are there. And we plate them and do all this. And this, we actually do in the genetics lab, they do a lot of this type of stuff. We use this kind of velvet stuff to plate them and all that kind of stuff. So I want to go through all of these things. I don't want to do it. This is what I want to tell you about. You remember I mentioned the word backwards mutation or reverse mutation. It's very rare. It's very hard. Imagine how powerful a mutagen would be if it could change something that's already mutated back to normal. That's exactly what the Ames test is about. Cancer, carcinogens, most of them are mutagens. They alter DNA. So, and it's because of this, they found a quicker and cheaper way, the FDA, to test the effect of certain chemical agents in microbiological systems. Mutagens increase low frequency of spontaneous reversions. In other words, it actually creates a backward mutation. The Ames test. They take a type of salmonella. Now, not all salmonella are nasty and evil, okay? But they take a particular salmonella that happens to require a chemical called histidine. Not that you have to know that, okay? And what they do, if the chemical is a mutagen, reversion rates increase relative to the control, meaning they take some of this chemical that's proposed for a new antibiotic, a new cosmetic agent, a new medication for blood pressure, whatever, and they mix it in such a way with this bacteria that's already mutated. In order to mimic what it does in us, they have to use some enzymes that come from an animal liver. So they take rat liver enzymes and mush that up with it all together. When they let it sort of percolate in that environment, sort of get together in that environment and grow for a while, what will happen? Then they check to see if it has changed that salmonella back to the regular type, where it doesn't require this histidine in order to grow. That is reversing it from a mutated form to the original, what we call the wild type form. If it does that, that chemical agent is a very strong mutagen and is almost assuredly going to cause cancer in animals. That is the aim state. That's something they're going to ask you about in a healthcare kind of, I would think in an exam, licensure exam, there would have to be some familiarity. So I need you to be familiar with it. It's a backwards mutation, and it involves testing it with an already mutated kind of salmonella and making it revert back to its normal stage. Got it? Good. Hence that. We're almost there. We get to show videos for the rest of the class, I think. Not those videos. I have other videos to show you. I'm not going to show you about the family trip and everything like that. Pictures from our vacation. Not that you want to see that. This is what bacteria do. The DNA can move around. DNA from dead bacteria can combine with living bacteria and change it. Dead bacteria, viruses can infect bacteria, rather, living bacteria, and actually, by accident, pick up some of the bacterial DNA and transfer it to another bacteria. And bacteria actually, by direct cell-to-cell contact, this is the one we use. And this is the one we refer to. We use a variation of this for genetic modification, but the one I refer to mostly, this is why a Staph aureus can become MRSA overnight. If there's one MRSA sort of in the mix, it can change a whole population to MRSA overnight. You don't need a lot of mutations. They have interesting names called transformation, okay, which involves naked or DNA from a bacteria that has died and fragmented. That's one, naked DNA here. Transduction, where a virus actually acts as the vector or the transport mechanism. It's not a real virus, it's pretty much the capsid of a bacteriophage virus. And lastly, and the one that occurs most, these are in order of frequency, least to most. Conjugation, which involves something I asked you about previously, which is an F. pilus or pilus, okay? That protein appendage that allows DNA to transfer. I think you remember the question. There's a reason why I put certain questions in, see? And I'm still super, I thought you all came here at 9 o'clock in the morning because you were so anxious. Having had a class at 8 right upstairs, this is bothersome. And I thought it was all because I was, we got to hear his lecture. It's so fun and fascinating. No, we're here anyway. We'll show up. I'm not going to show you the pictures. Not going to show you that. Transformation. DNA not within the cell of the virus. Cells that have burst and given it off. There's chemicals we use in the lab sometimes called DNAase. Okay, not that you have to know about that. We talk about competency. Not every cell can do this. When we do this in the laboratory, and they don't do it you could do it in a microlab, but we don't do that. We do it in the genetics. It has to do with heating it and chilling it and using calcium salts and stuff like this. Let me show you the very, very famous test, and then I'll get to the animation. 1920, more than a century ago. This researcher named Griffith was studying pneumonia, and he was growing it in the lab. The most common type of bacterial pneumonia is Streptococcus pneumonia. About a third to a half of you have that in your throat anyway. The difference is that the Streptococcus pneumonia, that's the bad boy, has a capsule. Remember the recurrent theme, encapsulated bacteria very often are associated with giving your immune system a hard time because they can't be gobbled up and destroyed by a macrophage. So he had these encapsulated Streptococcus, okay, and he had two types. He had ones that were encapsulated, ones that weren't. And what he did, he didn't know about if it was DNA. He didn't know if it was a protein. All he knew was that when I injected this into, this is not great for the mouse. When he injected it into an otherwise healthy mouse, the encapsulated ones caused the mouse to die. When he injected them, the ones that were non-encapsulated, the mouse had no effect. So he was trying to figure out what is the difference. Well, I'm going to heat the ones that are encapsulated. Effectively, he reasoned that that would have a good chance to destroy whatever was killing the mouse, and it had no effect. Okay, in other words, it would satisfactorily remove the thing that would kill the mouse. Then he said, well, I'm not sure. Now, what if I mix the ones that are encapsulated with the ones that are non-encapsulated? Since this, separately, they didn't kill the mouse. But when you put them together, the mouse died. And what did he culture out of their lungs? The encapsulated ones. He reasoned there was something in those dead cells, not knowing it was DNA, that caused the cells to change and form a capsule and become deadly. Cause pneumonia, because that's what the mouse died. Brilliant. It wasn't until we had radionuclide imaging, until we could get radioactive markers, if we didn't know if it was protein or whether it was DNA. And there was a very famous experiment in 1960, that you don't have to know the name, called Hardy and Chase, where they eventually proved this, and then even another one in the molecular biology to prove, to even give it more proof. 100+ years ago, he knew there was something. That's called transformation. So this is a process that goes on. Bacteria, large populations of bacteria die all the time, particularly if, okay, they're subjected to various kinds of chemicals and or antibiotics. Let me show you the animation. I will try to send these to you individually or in a file that you can open up. If you cannot open them up from this thing, which is over here, these animations. And so we'll look and see what they are. I want to make sure I have the right one. Some of them are a little bit more weird than others. I use the MP4. I'll
into a recipient cell. In the first step, double-stranded donor DNA binds to specific receptors on the surface of a competent cell. One strand of the donor DNA is degraded by nucleases while the other strand enters the cell. The single-stranded donor DNA pairs with an homologous region on the recipient DNA and is integrated into the recipient genome by a breakage and reunion mechanism called homologous recombination. If there are any differences between the nucleotide sequences of the donor and recipient DNAs, the mismatch repair system comes into play. The repair system removes either the donor or the recipient strand and replaces it with the complementary sequence. Since either strand may be repaired, some cells contain the new donor DNA and others have the original DNA sequences. In the laboratory, cells are plated on selective media so that only the transformants will grow.
And so, effectively, what has happened with all of this? Just to run it back a little bit. It's there, even without the sound, so to speak. Yeah, play again.
DNA transformation involves the transfer of naked DNA.
And so what happens is, this is typically a cell, this is naked DNA from another cell that has died. It's very specific to receptors. The cells have to be receptive to it. That's a fancy term called competence. the big term to remember here, and it's neat because the DNA doesn't just jump on at the end and cause another massive mutation. It's called homologous recombination. Recombination is a fancy term, okay? That means old DNA is being replaced by new DNA. Okay, so it either could be added to, but homologous means it goes right to the spot it would have come from normally. And it kicked the other guy out, and it degrades. No idea, no explanation, but do remember the name homologous recombination. Moving on, and this is the mismatch repair. We just let it go for a while. And so as it replicates, and they're going to show you, type A and type B, it says likely to, and this is mismatch repair. It can remove the mutation or remove the one that's supposed to be there. That's why it's roughly 50-50. And again, we do this, and we have a kit in the lab, comes from Carolina Supply. Everybody uses it. They use fluorescent markers. We do it in the laboratory. We do it with genetics. It's done in high school labs. So a lot of times I'll have an AP biology, depending on how the budget, so to speak, of the lab, depending on the school. I know that, like my kids went to Fox Chapel, which is a pretty wealthy school district. They did, but they had a really great teacher. Let me rephrase: not my kids. Never, none of them have ever been in a science laboratory, other than mine when they come to visit. It's okay. It's very frustrating. Dad, explain this to me, right? So that's really the saga of this. Not much more to say about it. See you later. Okay? That was relatively brief and hopefully painless. Moving on. Next thing on Hit Parade. Similar and occurs more frequently, bacteriophage, these viruses that don't affect us directly, but affect bacteria and invade bacteria, are very interesting viruses. Again, the nickname is phage. What they do, and remember, it's all they do is inject the nucleic acid, and then the enzymes that are part of it start chopping up the bacterial DNA and effectively take over the cell. We're going to talk about, you're going to see an illustration for what's called generalized transduction. It's easier than looking at the illustration for me to explain it to you by way of the animation. And again, I'll let you listen to it, and then I shall endeavor to try to clarify it a little bit, because all of this is kind of new. The part that you need to remember, the virus is really picking up a piece of DNA that's in the process of being destroyed and latches onto it. And because remember, viruses work differently. They take over the cell, they create a little assembly line, they make more DNA or more RNA, they make more capsid proteins, they make protein spikes, and basically the cell, in this case, the cell bursts. So I'll show it to you, and this is called, and there's something where it goes to a particular location, but we're going to do the generalized transduction. I think it's the easiest one to explain.
Carried from one bacterial cell to another by a bacterial virus called a bacteriophage, or phage.
You start from the beginning again.
The phage attaches to the bacterial cell and injects its nucleic acid into the host cell.
Give me a hard time, though.
In generalized transduction, a segment of DNA is carried from one bacterial cell to another by a bacterial virus called a bacteriophage, or phage. The phage attaches to the bacterial cell and injects its nucleic acid into the host cell. A phage enzyme is produced that breaks down the host DNA into smaller fragments. Phage DNA is replicated and phage coat proteins are produced. During formation of the mature phage particles, a few phage heads may surround fragments of bacterial DNA instead of phage DNA. The phage particle carrying the bacterial DNA infects another cell, transferring the bacterial DNA to the new cell. When the bacterial DNA is introduced into the new host cell, it can become integrated into the bacterial chromosome, thereby transferring genes to the recipient. This cell then multiplies and carries new genetic material.
And so and I'll give it to you this way, and we'll start over here. I'll take off the sound. So as we begin to look at this process, the whole idea, this is, if you'll notice, they're demonstrating, this is the DNA from the phage. This is the host. They did red and blue. Again, it's not a random. It has to go to a specific receptor, and the cell has to be receptive to it. And so basically, this is how a virus invades a bacteria. And what happens is it starts to break apart and eventually destroy the DNA. But some of the DNA fragments survive and are inadvertently, because the virus is just, it's a process where it incorporates, as you can see here, bacteria. So really, it's not a true virus, but the virus apparatus is a means that potentially could convey it to another bacteria cell. It's something that happens. If this happens 1 out of 100,000 or 1 out of 1,000,000 times, you and I would look at each other and go, well, that's pretty rare. You see in the lab all the time, how long does it take to get those little dots on your plates? Each one of those dots is 1,000,000 bacteria. I mean, if whatever you put in the lab yesterday, Tuesday, or let's say you did it on Tuesday, you go back in the lab, now you're going to have stuff growing everywhere. You're going to have millions, if not billions, of bacteria growing. So when we think tremendously large populations, so these numbers are significant. A rare occurrence in us might be a common occurrence that occurs in bacteria. I say it all the time, it's a numbers game. It's all about the numbers. So moving on with this, what happens? It reaches what we call a burst size. And when the darn thing bursts, like that, now effectively all it is a vector, conveyance, to bring this DNA that might have resistance, might have some other, make it more virulent, make it more nasty to you or I, or to another species. And again, once it does that, effectively, once it's in there, homologous recombination, just like transformation, and it's subject to mismatch repair. So that doesn't change. So it's not, understand that it's a very similar process. And even though it happens more frequently, but we don't have the concept of how frequently bacteria mutated and how they become mutated. Which brings us to the one that is most common and probably best understood nowadays. I think would be a fairer way to describe that than before. So you'll let me X out of this and X out of that. And it's easier if I explain it to you. This is called conjugation. And so originally, it takes the longest time to explain. Originally, it's over here. There's several types. We're going to do the F plasmid. Let's just watch it and listen, and we'll go from there.
Requires direct contact between donor and recipient cells.
They have to be in contact, and this is that F pilus.
Carry the F plasmid, enabling it to make an F pilus and act as a genetic donor. Recipient cells that lack an F plasmid are referred to as F minus. The F plasmid encodes for the F pilus, a protein appendage that attaches the donor to the recipient cell. The first step in plasmid transfer is contact between the donor and the recipient. The F pilus of the donor cell recognizes and binds to specific receptor sites on the cell wall of the recipient cell. The plasmid then becomes mobilized for transfer when a plasmid-encoded endonuclease cleaves one strand of the plasmid at a specific nucleotide sequence called the origin of transfer. A single strand of the F plasmid, beginning at the origin of transfer, enters the F minus cell. A complementary strand to the single-stranded plasmid remaining in the donor is synthesized via the rolling circle mechanism. Once inside the recipient cell, a complementary strand to the single-stranded DNA is synthesized. When F plus and F minus cells are mixed together, eventually all of the cells become F plus.
So that's why overnight, because not only do all the cells that have been altered become F positive, they go around basically conjugating with or transferring their fragment of genetic material to cells that are not. And eventually the whole population becomes F positive. So it really only takes one because, again, bacteria are constantly replicating and you're changing it. So if we just look at the process again without the sound, effectively what happens, this is called F positive because it has it. The only thing it means being F positive is it has the F plasmid, and that's the information to build the pilus and to accomplish the transfer. That's all that it means. It may or may not carry resistance. It may or may not carry the ability to make a capsule. Anything could be there. But at the very least, that's what it has. When they get together, and that's when they looked at this, anatomists thought this was some type of sexual, process in bacteria, but it's not. The reason is it's just transferring DNA. It's not, we're not really doing a mother, daughter, father, son type of a deal that's coming here. And because the other one, it really is not a form of replication per se, it's an altering. It starts at this thing called the origin of transfer. And this whole rolling circle deal means that, because remember, the cell immediately becomes F, it's no longer F positive if it loses the plasmid. So it's a continuous process. The outer strand, remember it's double-stranded DNA, identical, is being moved, and simultaneously it's being replicated. So once that entire strand moves through, and it just takes a few minutes, comparatively speaking. Okay, it's a relatively rapid process. Immediately, then it forms the second strand, making it now, and that immediately stops the process. As soon as that strand is replicated, it becomes F positive, assembles the F pilus, and it goes again and again. There's some funny changes when we start. We basically take a variation of this mechanism to make genetically modified products like human insulin, like Pitocin, all of those things. And so there's not a lot there. I don't have time to do the transpositions one. I will send it to you so you can take a look and see how rapidly. That's the whole one with the McClintock studies and the corn that we see so commonly. So we'll leave it there, and I'll make sure you have all the information because I'm going to be sending you another recording with some metabolic scheme information I'm kind of working on. If I'm ambitious, I'll try to get it out tomorrow.